Thanks, Jacques, for the introduction, and I would like to thank everybody for connecting to this webinar. I hope you will find my talk interesting and worthwhile your time. I will talk about our project that we started at the beginning of the COVID-19 pandemic about 2 years ago that uses neutron crystallography to inform drug design targeting a specific drug target of SARS coronavirus 2 that's called main protease enzyme. Before I start, let me give you a few biography facts. I studied in Kharkov State University. So originally I'm from Ukraine. I got a master's in organic chemistry there, a PhD in physical chemistry with Professor Philip Coppens in Buffalo, New York, working on metal organic complexes and photocrystallography that we developed in that lab. I switched to biochemistry later on as a postdoc at Georgia State University, and then my sight fell on neutron crystallography that I thought is a great technique to study enzymes, because enzymes move chemistry pretty much exactly as organic chemistry does, but with huge efficiency, and hydrogen atoms are very important in those reactions. So I moved to Los Alamos and then to Oak Ridge National Lab, where I'm currently an instrument scientist on both IMAGINE and MANDI protein crystallography instruments. I have more than one project. This project is just one of the several that I run. Some of them are funded by NIH. Currently, I have an NIH grant studying the vitamin B6 dependent enzymes. It's one of the most diverse family of enzymes, and a lot of them are implicated in disease, such as cancer, and also for novel targets for antibiotics. We also have a project on the reactivator design for nerve gas and pesticide poisoning of human acetylcholinesterase. And as much as the world tried to get rid of chemical weapons, even now we keep talking about them and their possible use in the world, which is really unfortunate. So to set the stage, let me first talk about the protein that we're studying. This protein is working during the replication cycle of the coronavirus 2 at the stage of proteolysis. So after the virus infects the cell, its genomic RNA is partly translated into 2 long polyproteins. And that's one of the very first stages in the replication cycle. And really nothing else happens until the main protease and another PL protease actually cut the long polyproteins into individual chains that form the replication transcription complex shown here. So with that, the virus is able to create the so-called nested set of subgenomic RNAs. And that's where most of the other proteins are produced for the virus to replicate. So if the main protease is inhibited, the replication cycle stops. So it's one of the most important drug targets that is being studied right now. So here's on this slide a nice movie showing how it works. Literally and metaphorically, this enzyme is the heart of the coronavirus 2. As you can see, ironically, it's shaped like a Valentine's heart, and it's absolutely indispensable for the life cycle of the virus. It cuts those polyprotein chains as shown here. If we create a very good small molecule inhibitor that binds tightly, the enzyme function is stopped and the virus replication cannot proceed. Hence, a lot of interest in these studies. So here's the protein structure itself. So I'm gonna go into a bit of detail because we're looking at hydrogens and we need to know a lot of details about the active site. This enzyme is homodimeric, so there are two identical peptide chains. Each one has 3 different subunits. The green and blue are so-called catalytic subunits. The orange is the helical subunit, and it's very important for the dimer formation. The mature enzyme, the active form is homodimeric. So this dimer has to form for the protein to work. This enzyme is also a cysteine protease. So in the catalytic site, there's a cysteine residue, catalytic cysteine, catalytic histidine, forming a dyad. But unlike many other cysteine proteases and also serine proteases, coronavirus main proteases do not have a third catalytic residue. Instead, there's a conserved water molecule shown here that's actually surrounded by a few other residues, aspartate, asparagine, and the tyrosine 54. So these residues, together with the oxygen, with the water molecule, form the so-called partially negatively charged cluster that has been shown by other work acts as the third catalytic residue. So it's a very important part of the active site. You can see also there are more histidines in the active site. So biochemists know that histidines are ionizable residues. They can be neutrally charged, so 0 charge, or they can be doubly protonated with hydrogens on, say, on both nitrogens. Then the histidine becomes +1 charged. And in this case, we have 4 of them here, plus cysteine that can be protonated, deprotonated. When it's deprotonated, it's negatively charged. So this active site has an opportunity to have very interesting electrostatics. These residues are ionizable. They're also titratable at physiological conditions, which means that they can have different protonation states depending on the environment. Either it's ligand-free form of the enzyme, or maybe substrate binds, inhibitor binds, which inhibitor binds. These protonation states can change. And this is important to know. Those protonation states have to be known for drug design. And especially these days, with the invention of theoretical tools and a lot of drug design moving into the high performance computation, computer models have to be accurate and correct for best results. So if we know where the hydrogens are, we can provide computational scientists with the correct and accurate distribution of charges and electrostatics and improve their modeling. The other important part of the active site is how the inhibitors and substrate bind. So here I'm showing a little PyMol movie where we will see the substrate binding subsites that are extremely important for drug design. So here's the substrate that's bound to a mutant enzyme that we made, where the cysteine, catalytic cysteine, was substituted with alanine so that we can bind a substrate and it is intact. We can see how it binds. Here, glutamine in this place is binding in the subsite in brown. That's called substrate binding subsite S1. it's lined up with the with these residues 140 through 142. Glutamate 166 is extremely important for the formation of this site, and this site is very interesting. First of all, it's pre-organized, and its shape is the same whether it's a ligand-free enzyme or it's a ligand-bound enzyme. It's also the most selective subsite in this enzyme. It exclusively binds glutamate. So for drug design, everybody has been trying to create chemical groups that actually mimic this glutamate. And you will see throughout my talk these groups that are similar to this glutamate. It's a selective, pre-organized site that's most important for drug design. And this histidine here, which is called 163, will become extremely important for drug design throughout my talk. So then we have the other site, which is called S2. It's nearby. Here, this leucine is binding in the subsite S2. As you can see here, with the pink, it has a few residues lining it up. The most important one is methionine 49. It actually, in the ligand-free form, it blocks this site. So this site is not really realized before inhibitor binds, but it's a very important one. It's a hydrophobic site. Then we go to subsite S3, S4, and S5. And here you can see S3 site where this valine is located. It's actually not even a subsite. It has no borders. It's on the surface, fully exposed to the solvent. So for drug design, it's kind of weird to find which groups have to be here. And where this alanine is subsite S4, another important subsite. This leucine 167 actually blocks it before an inhibitor or substrate binds. So now it's carved out, while the subsite S5 to the right is also on the surface. It's lined up by this long S5 loop, but really does not have any cavity. So as I mentioned, we really need to know the protonation states of the ionizable residues in the active site. So if you look at this ChemDraw picture and you calculate simply how many combinations of protonation states and tautomeric forms of histidine we can come up with, we get to 108. And I argue that it's near to impossible to figure out which one we will see exactly with experiment, which one actually occurs naturally. Also, which one will be there when an inhibitor binds. Those are all open questions, were open questions when we started this project. And with X-rays, as shown on the right, the electron density gives you exactly the same picture for neutral histidine or protonated histidine, for example, while with neutrons, if we look at the nuclear density, we can clearly distinguish between the neutral histidine and both of its tautomers when the hydrogen is on this nitrogen or on this nitrogen, or we can see clearly the protonated histidine. So our first neutron structure was of the ligand-free enzyme, and the the nuclear density of the active site is shown here with the dark mesh. So what's important, the important observations here that we saw, one of them was really unexpected and quite interesting. It's the fact that histidine 41, the catalytic histidine, is doubly protonated, so it's +1 charged, while cysteine is deprotonated and it's -1 charged. So in this state, the catalytic dyad is in zwitterionic charge separated state. And this state is a really reactive one. So the cysteine is ready to attack a substrate or bind with a covalent inhibitor that you will see at the next part of the talk. Histidine 163 that I said is extremely important. It's found in subsite S1, is neutral. Here, 164 is protonated, and we can clearly determine the orientation of the catalytic and conserved water molecule. So this sets the first look at the protonation states of this enzyme. This was at the time the very first neutron structure of any coronavirus protein and of any cysteine protease, which was pretty cool. We then went on to get a neutron structure with a covalent inhibitor, because we want to understand to inform the drug design how these inhibitors bind. Here we chose telaprevir, which is shown on the left. This is one of the older clinical drugs that was used to treat hepatitis C infection, and it's a specific hepatitis C protease inhibitor. Turns out that these inhibitors bind well to the main protease of SARS coronavirus 2, and our lab and other labs actually did some work on these inhibitors, including later drug design using them as the basis that I will show later. This inhibitor is covalent. It has a warhead, which is carbonyl here, shown in red. When it binds to the enzyme, cysteine attacks the carbon and creates this covalent adduct, which is called hemethioketal. It works extremely stereospecifically, so that only one diastereomer is formed, as shown here with the nuclear density. We then looked at the protonation states in the active site. We see that the histidine 163 becomes protonated. And this is an interesting observation, because this P1 group that binds in the S1 site is hydrophobic. It actually cannot form hydrogen bond with histidine. It's not really a specific group for the inhibitor, but histidine 163 becomes protonated and it recruits a water molecule from the solvent. So we thought that this is an intrinsic property of this enzyme for this group to be protonated. And the conclusion from here is that an inhibitor should have a group, instead of this hydrophobic chain, it should have a group that is capable of producing a good hydrogen bond with a protonated histidine 163. Histidine 164 and 41 are deprotonated, and in fact, if you look here on the lower left side, the histidine actually donates its proton from the ligand-free structure to this oxygen, and it becomes OH, and it becomes neutral. Histidine 172 we saw deprotonated, and this is one that we're not gonna even talk about later on, because it turns out that this residue is locked by this nitrogen to a main chain NH, the main chain amide NH. So it's not really titratable. It will never get protonated in the physiological conditions. So these are the charges. What's interesting is that there's only one +1 charge, and that seems to be a feature of this enzyme. In this little movie, I'm showing how the electrostatics in the active site changes. So the red surfaces show the positive charges, blue is negative charges, and gray neutral charges. So you can see how when the inhibitor binds, there's redistribution of electrostatics changes in the protonation states. So the electrostatics of the active site changes. It's something that we think is extremely difficult to predict with computation. You can also notice that the active site kind of opens up this little helix on the right and this beta hairpin loop. They move. So that's a very important feature for this enzyme as well, and the one that's difficult for drug design. And in this movie, I'm showing exactly that. As I mentioned, subsites S2 and S4 are not really preformed before a substrate or inhibitor binds. They're blocked by residues, and that's why we call them cryptic subsites. Means that in the apo or ligand-free structure, they're not there per se. So once the inhibitor binds here, you can see how the subsites are formed. So look here and here. Once inhibitor binds, there are cavities that appear for these inhibitor groups to go in, and they will disappear once the inhibitor dissociates. That's a feature that complicates drug design, especially complicates drug design using computational tools. So in this part of the project, we found out that main protease is protonation states are tunable, that overall electrostatic charge is maintained at +1. And it turns out that that seems to be the property of the enzyme. Every time we get a new neutron structure with a different inhibitor, the overall electrostatic charge in the active site remains +1, even though all the individual charges may change. The active site conformation dynamically adapts to inhibitor properties, and that, which we call plasticity, with the presence of cryptic binding subsites, presents challenges for inhibitor design and in silico modeling. So in the next part of our project, we did the so-called structure activity relationship, or SAR, study for non-covalent inhibitors to specifically look at subsites S1 and S2 and understand the properties that inhibitors should have to have good binding properties to this enzyme. This work started with collaboration with scientists from Argonne National Lab, who developed a supercomputer high throughput screen of non-covalent inhibitors. They took a library of 6.5 million ligands. They created their methodology so that we actually can overcome those cryptic subsites. So they used a lot of different conformers of the protease. It turns out that they could create 336 billion of those conformants and performed a lot of docking and molecular dynamics simulations. A number of compounds were selected by these computational simulations. Obviously, most of them did not work in experiment, as it always happens, but one actually was active, and its chemical structure is shown here on the lower left. It has a P1 group, which we call a uracil-like group. It's the group that binds in the subsite S1. That's where we know that histidine 163 is sitting, and it turns out to be one of the most important residues for drug design. There is a linker, which is called piperazine, and it binds across the catalytic residues. and there's a P2 group, the dichlorobenzene ring that goes into the S2 subsite. We grew these beautiful crystals, got a room temperature X-ray structure to make sure that it binds, and also compare to the computation. How do we predict it binds and how does it really bind? And on the top right, you can see the poses from experiment with yellow carbons, from prediction in yellow carbons, and from experiment in cyan carbons. You can see that prediction is pretty good, except the P2 group is a little off and it's flipped 180 degrees, but that's not a big deal. The computational tool actually worked really well. So now with this information that we got, we decided to obtain a neutron structure for this compound, because we wanted to do this structure activity relationship study, and we wanted to inform it by knowledge of the electrostatic environment and the protonation states inside the active site. So here's the nuclear density map and the OMID density map in violet. One of the most interesting observations is that the catalytic histidine 41 is neutral, as shown here. Its conformation is actually flipped 180 degrees compared to the inhibitor-free structure. The cysteine is protonated, the catalytic cysteine. We can clearly see deuterium. So it's a very nice observation. Everybody who has done neutron diffraction knows that sulfur has very low scattering power, so that in many cases the density is very weak for cysteine. So we were really asked and criticized that maybe in the inhibitor-free structure, our observation of deprotonated cysteine may be incorrect because of low scattering power of sulfur. But this observation actually reinforces our conclusion that we can see the protonation of cysteine if the data is good. We can see when it's deprotonated, we can see when it's protonated. Histidine 163 is also protonated. So as I said, it's an intrinsic property of the enzyme to change the protonation state of 163 from neutral to protonated when inhibitor binds. It has a really good hydrogen bond to the uracil-like group, and that hydrogen bond becomes extremely important for drug design. And another cool observation here is that the nitrogen in piperazine is protonated. So the whole little small molecule inhibitor is actually a cation. It's +1 charged. And what's interesting about it is that this is not a permanent charge. So it's in equilibrium with neutral and protonated form. And that can become important for drug design because a neutral form would cross biological membranes, the protonated form would not. So if you have both, it's important for drug design, for later drug design. We also looked at the rest of the active site. So here's our histidine 41, the catalytic water molecule. Histidine 164 is neutral again, as in the previous inhibitor structure, but it's a different tautomer. So now we can see that the active site can not only change protonation states of these histidines, they can actually form different tautomers, so that the hydrogen here is on a different nitrogen than before where it was here. And with that in mind, we continued with the SAR study. We thought that now we have all the information needed to do a good structure-activity relationship, because that requires modifications to the chemistry of this ligand. it requires modifications that are, first of all, possible to synthesize, and second of all, are informed by our structural knowledge. And here's the movie of the crystal that we used for the neutron structure in the polarized light. It's pretty cool. We also used virtual reality that is shown here. So with virtual reality, and specifically it's the Nanome software, you can go inside the molecule and in real 3D examine its structure. You can use the tools inside this software to actually build new molecules. And the software will tell you if your created molecule is fitting in the active site, or maybe it's clashing with the residues, so that you can not only analyze the structure, you can also minimize the number of molecules that you need to synthesize so that your SAR study is most informed. It's not overwhelmingly huge, but you learn what you need to learn. And here are the structures that we made. At the top, you can see that we have changed substituents on the P2 group on this benzene ring. We chose different ones, but with the help of VR and also with the knowledge of what can be synthesized, we chose a number of them. We also changed this nitrogen here to carbon. As you can see on the left, the lower left side, there's no nitrogen there. So now we completely removed the possibility for this molecule to get protonated and to be a cation, which is important to understand in terms of binding affinity. We also created a compound that's shown here, actually two different enantiomers with this bond in the uracil-like substituent being hydrogenated. So now we have two sp3 hybridized carbons. So it actually is bent relative to how its conformation is usually in the other molecules. So this particular molecule actually tests our prediction whether the hydrogen bond to histidine 163 is important. When we synthesize those, we found very interesting trends, and some of them are shown here. The parent compound is on the left, and it has the IC50 of 0.7 micromolar. So IC50 is the inhibitory concentration that reduces the enzyme activity by 50%. We measure it in solution. The lower this number, the better the affinity of the inhibitor to the enzyme. So we really want this number to be as low as possible. For clinical drugs, it has to be nanomolar or lower. So when we changed, for example, introduced another halide, this didn't do anything to the affinity. So it turns out that that site actually likes halides. When we replaced chlorine with, say, CF3 group or aldehyde or even alcohol, we noticed that changing to a more electronegative group and a little bulkier group actually reduces affinity, maybe a little bit here, but by about 10 times for this compound and for this compound. When we looked at the compound where the nitrogen is replaced with carbon, so there is no way this compound can be protonated and cation, you can see IC50 shot up more than 10 times. So the ability to this ligand to be protonated is extremely important for the small molecule affinity and binding to the active site. We also found out that if this double bond is a single bond, that also reduces the affinity of the inhibitor to the enzyme, and actually much more than 10 times. So we also learned that really the hydrogen bond, the proper hydrogen bond to histidine 163, which is protonated, is absolutely essential for drug design. You cannot have a good inhibitor without forming that hydrogen bond. We looked at the thermodynamics of binding of several of the best inhibitors using isothermal titration calorimetry. So what's interesting about the calorimetric measurements is that you directly measure binding and you get the enthalpy of binding, entropy of binding, and obviously overall delta G. So you can distinguish between what's driving the binding. So if you can look at the delta H for these three compounds, we found that HL368, where there are 3 chlorine atoms, very similar to the initial one on the left, but turns out to be about twice as good. This one with the CH3 group in the para position of the benzene ring is about the same as the parent compound. So what's important is that we find that these inhibitors are enthalpy driven. That's one of the most important things for drug design. The best inhibitors out there, the best clinical drugs, are enthalpy driven binders. You do not want just a hydrophobic effect or entropy to be the driving force for the inhibitor binding. But for these inhibitors, what's interesting was that you can see that delta S for the parent compound and for the third one is actually negative, which is unfavorable. While just changing one group to chlorine, we reverse that situation and the delta S is slightly positive and it's favorable. So you have to balance between the delta H and delta S, but you cannot have a situation for a drug where delta S is unfavorable. And for this enzyme, this is quite an interesting observation. We did not expect it, and we think that one of the reasons is that the active site of this enzyme has very few water molecules, even in the inhibitor-free structure. So when the inhibitor binds, there's not much entropy coming out from removing the water molecules from the enzyme surface. So in this part of our study, we learned that one atom difference can significantly alter inhibitor binding affinity. For the first time, we used VR for drug design that allowed 3D structural analysis and inhibitor building that helped tremendously. This allowed us to tailor the inhibitors to binding site and minimize the number of those inhibitors that needs to be synthesized. And the third part of my talk is about covalent inhibitors. So here we actually did real drug design, and this part is really one that I like the most. I think so. We used the hepatitis C virus protease inhibitors that I mentioned at the beginning of the talk, and we created hybrids out of them. So here they are: boceprevir, nirmatrelvir, and telaprevir. So we saw telaprevir, boceprevir, and nirmatrelvir were looked at by us at the beginning of the pandemic, and by some other groups that decided to look at these inhibitors as potential inhibitors to reuse for the treatment of COVID-19. Of course, boceprevir showed some good activity, but was not any good to be used in the clinics. But the structures of these compounds can be used to create new ones that are more tailored or more specific for the main protease of SARS coronavirus protease. And that's what we also did. So here I'm showing what we actually did, boceprevir, nirmatrelvir, and then there's this one that's called GC376. It's an older drug that's been studied in the clinics to treat feline peritonitis virus. It's another coronavirus that affects cats. One of the crazy things about it, and we are so lucky with coronavirus 2, is that the feline peritonitis virus has 100% mortality. If cat gets infected, it's dead, period. So we're really lucky with SARS coronavirus 2 in that regard. So what we did, we changed this P1 group, which is obviously hydrophobic in the hepatitis C protease inhibitors, with this group, which is called gamma-lactam. It's the group that can clearly form a good hydrogen bond to histidine 163. We also looked at replacing those P1 prime groups that are amides here, and also changing the warhead. So the three compounds that we created are called BBH1, BBH2, and NBH2. In BBH1, you can see this is a boceprevir-based hybrid inhibitor. P1 prime is benzothiazole. The reason we put it there is because in earlier studies on SARS coronavirus 1, some inhibitors were designed with this group. So we decided, well, we want to look at how it performs as well. In this compound, we have carbonyl group here, which is the warhead, while in the other two, so BBH2 is as well boceprevir analog, but there's no P1 prime group, and the warhead here is the nitrile. So these covalent warheads have been looked at previously for SARS coronavirus 1. So we decided to look at that as well. And the NBH2, you can see it's basically to the left of this gamma-lactam, it's nirmatrelvir. To the right, it's gamma-lactam and the nitrile group. So we synthesized those and set into trying to understand how they bind, how they actually work, and whether they have antiviral activities. So first, we obtained a neutron structure of BBH1, and the hydrogen bonding is shown here on the right-hand side. This one is interesting because it has the carbonyl as the warhead. And when, like in telaprevir, when the carbonyl binds to sulfur, produces hemithioketal. And from the very beginning, we were thinking, well, if this oxygen here is looking into the so-called oxyanion hole, you see those all NH groups that form the oxyanion hole that should stabilize the negative charge on oxygen. The question has been, is it protonated? Is it negatively charged when these compounds bind? And on the left-hand side, you can see the electron density in red and the nuclear density in violet. So the structure was very good, and let us look in much detail about how this inhibitor binds and what the protonation states are. So on the upper left, I'm showing the nuclear density around this oxygen, this hemithioketal conjugate. You can see it's hydrogen bonded to one of the NH groups of cysteine 145 in the oxyanion hole. It's also hydrated by a water molecule. There's another possible very weak hydrogen bond here. And most important observation is that it is negatively charged. There's no deuterium atom found. So it's truly O- oxyanion, and it's stabilized inside this preformed oxyanion hole, basically mimicking the very first intermediate in the catalytic reaction. So that's a very neat observation. For the P1 group, which is the gamma lactam on the right-hand side, you can see it forms a really good hydrogen bond with histidine 163, and it's extremely important to have it there. While in the other part of the site, something very interesting and unexpected happens. And that's because there's this weird benzothiazole that previous designs used for SARS coronavirus 1. What's weird about it is that it's huge. It's huge and it's flat, and it has basically no conformational flexibility. So once this compound binds, this big fused cycle crashes into the histidine 41. So what happens is histidine 41, which is catalytic residue, is pushed away to the right. It actually flips 180 degrees. It becomes neutral. There's only deuterium here, as you can see. And as you can see, there is a direct contact to histidine 164, which is also neutral. And the end effect of this cascade is that the catalytic water is pushed out of the active site. There's no catalytic water in this active site. So that's a dramatic change to the structure. We also learned that benzothiazole is not a good substituent. It makes the molecule, the compound, very insoluble. So it was hard to work with it in solution, but the neutron structure is a very interesting one. We also obtained room temperature X-ray structures of BBH2, NBH2, and here on the bottom right, you see the Pfizer's compound. Probably many of you know, this is the now clinical drug that Pfizer designed. It's called nirmatrelvir. It was approved in US by FDA to treat COVID-19. It's a very effective drug with the effectiveness of more than 90% to avoid hospitalization and death from COVID-19. You can see that its design is very similar to ours. This portion here, this fused ring and these third butyl groups are exactly the same as in boceprevir. So it turns out that Pfizer also had the same idea and did the design of hybrid inhibitors. Instead of these hydrophobic groups that we have to the left, they used the CF3 group. That's a very interesting design feature. According to Pfizer's paper, they put the CF3 group there for metabolic stability of the compound and for better penetration through the biological membranes. But they never mentioned the other cool thing that we noticed with our room temperature structure. As shown here on the bottom, the CF3 group forms this very strange F-oxygen interactions. It turns out that these interactions are favorable, and those are so-called closed shell multipolar interactions. Even though both atoms are quite electronegative, the multipolar interactions are favorable. And that actually helps this compound to be quite a good inhibitor, which is seen here when we did the isothermal titration calorimetry measurements. This directly gives us the affinity, which is called KD, the dissociation constant. You can see that the Pfizer's compound has 7 nanomolar affinity, so it's really good right there to be a clinical drug. But our compounds are not far behind. The BBH2 and NBH2 have about 26 to 30 nanomolar, which is about 100 times better than the initial hepatitis C protease inhibitors. So by changing those a couple of groups, we made a leap by improving affinity by 100 times. We also see with the enthalpy, the Pfizer's compound is mostly enthalpy driven to bind to the enzyme with very little entropy. And this difference in enthalpy of about 2 kcal per mole relative to our compounds, I think can be attributed to the fact that the CF3 group makes those very unconventional FO interactions. In the case of our compounds, there's also quite good delta S of binding. So entropy of binding is quite good, in much higher than Pfizer's, maybe because our compounds have more hydrophobic residues, hydrophobic groups. So that's our suggestion. But in the end, we created very nice new small molecule inhibitors that can be used for later design. And obviously, we collaborated with an antiviral lab in Memphis, Tennessee, to do antiviral measurements. So here you should actually look only at the middle column. These are the EC50 values. These are the effective concentration that reduces the potency of replication by 50%. You can see that for Pfizer, it's 250 nanomolar. Our best compound, BBH2, is about 880 nanomolar, which is way below the micromolar level, which is considered an important threshold for a good antiviral inhibitor. So with this study, we created not only good inhibitors, but actually antiviral compounds. And now we have a good basis to maybe design even better compounds. So in this part of the talk, we found that hybrid inhibitors are fruitful path to clinical drugs. Again, protonation states adapt to specific inhibitor, active site geometry adapts to inhibitor steric size, its electronic properties, and these hybrid inhibitors are conceptually superior to previous drug designs. To end this talk, I would like to say a few words about our future directions that we think about. Obviously, it became apparent from everything I said that we are using covalent hybrid inhibitors as lead compounds for further design of better main protease inhibitors. We are working to diversify the chemistry of the non-covalent inhibitors, where we did the structure-activity relationship study to improve their binding and to actually generate a lead compound that is more drug-like. We are studying main protease function, catalytic mechanism, and mechanism of maturation. As I mentioned, the very first stage of the replication cycle is the production of long polyprotein chains. Well, one piece of that polyprotein chain is actually the main protease. So what it needs to do first, it has to mature. So it has to cut itself out with the process that called autocleavage out of this long polyprotein, and then go on to cut other parts of it. So that process is maturation, and it's really not very well understood, or really at all. It's not understood how that happens. We also studying the main proteins dynamics with inelastic neutron scattering in combination with MD simulations. So that's where we do the so-called sub-terahertz picosecond vibrational dynamics studies. These picosecond vibrations are extremely important for drug binding because they are the ones that are included in the free energy of inhibitor binding, or they contribute to the free energy of inhibitor binding. And at the end, I want to acknowledge everyone that has been involved in this work. Daniel Caneller used to be my postdoc. He is now at the New England Biolabs as a scientist. He did most of the work, really great postdoc. All the papers we published were done just because of him. Leighton Coates was the instrument scientist on MANDI at the time before he moved on to the second target station at ORNL. So he helped a lot with data collection on MANDI, and Helen and Malcolm are scientific associates on MANDI and IMAGINE. We collaborated with ORNL Biology. Stephanie did a lot of solution measurements. So for example, those IC50 values were measured by her. Peter and Hui Li, they are synthetic chemists. So we are collaborating with them there at the Center for Nanophase Materials Sciences at ORNL. So they are synthesizing the small molecule compounds. Those two guys, Arvind and Austin, are from Argonne National Lab. They developed the high throughput screening techniques. ORNL Center for Structural and Molecular Biology helped us with the protein production, protein deuteration. My collaborator, John Louis from NIH, he's the big expert in isothermal titration calorimetry. And on the, actually on the proteases. And of course, I cannot leave out Matthew Blakely. He's been great collaborator and friend over the past 15 years. We worked together all the time on LADI, doing extremely good experiments and science. And for this one, he collected the data for BBH1 compound, which I can confess would not be possible to do anywhere else except on LADI. So this is great. And also I want to acknowledge the partially funding from the National Virtual Biotechnology Lab that US Department of Energy created at the start of the pandemic. And thank you everyone for your attention. I will be happy to answer any questions.
So thank you, Andrei, for this very interesting talks. So I don't know if we have some questions or comments. So Anne, I try to give you the floor. And you can talk, but you are mute.
You hear me?
Yes.
Yes.
Okay, thank you very much for your talk. It was really interesting and demonstrated very well the need of support labs and screening capacities, even if we do low throughput experiments at the end in the neutron world. I had a question about the first part of your first part, about the two cryptic sites.
Mm-hmm.
I talked about you. In the way you said things, I had the impression that you suggested that the drug binding would create these sites. Is that what you mean, or do you think there is a kind of a dynamic and the binding selects a conformation which has these sites?
Yeah, that's a very good question. So far, we we saw that these sites are always blocked by residues in the inhibitor-free structures, and also even in MD simulations, we didn't see them to open up completely to create the whole volume of those sites. That's why we think that it's an induced fit, and those inhibitor groups or substrate groups need to push the residues away to create the full volumes of those sites, subsites. Yes.
Okay, thank you.
Thank you, Anne, for your question. I don't see. Paul.
Just a quick question. Are any of your designed inhibitors under patent? You know, did you get to the stage where you had to patent anything?
Yes, we submitted provisional patents for both studies, for the non-covalent inhibitors and also for the covalent inhibitors. Those are new provisional patents. So the question now is whether we will go with full patent applications. That's an open question right now.
So would it be DOE that owned the royalties on, you know, if anything comes of it, you know?
I think so, yes. Yeah, because ORNL would be the submitting body for the grant for the patents.
But you don't have direct collaboration with a pharmaceutical company for this kind of research?
No. We actually tried to contact Pfizer several times, but that was unsuccessful. But you know, Pfizer is a multi-billion-dollar pharmaceutical company. It's hard to excite them sometimes. Plus, US government rules are pretty difficult, so it may be very difficult to even create an agreement to do this.
I think so. We have a question from Giovanna Fragneto. I give the microphone to Giovanna.
Okay, thanks. So nice talk. I just a curiosity. I was wondering about the other activities going on at ORNL with COVID-related science. So in solution with SANS or membrane work. Do you have sort of a group working a little bit in this area with the different techniques?
Yeah, I know that Hugh O'Neill's group at the CSMB, they've been looking at a number of proteins with SANS. I have personally not seen the results, and they haven't published anything yet. Some other people tried with the membranes and previously looked at some small molecules that like hydroxychloroquine that nobody wants to talk about any longer because those turned out to be wrong compounds to work on. But yeah, there are some activities not published yet.
Would there be mainly user collaboration activities, or do you have I'm not so familiar with ORNL, I apologize, I've never been there, but I just wonder if you have a group promoting work in life science areas.
Well, what we are doing, it's we are mainly driving this project as an internal project. For the SANS, there was a lot of also internal push for the COVID project. I'm not very much familiar with users for that.
Okay, thanks.
So we have two questions, one from Azisai. So I give you the floor for you asking your question. You are mute.
Oh, okay, thank you. Hi, Andre. That's a very nice talk. Thank you. Just a question on the last part. So what was your motivation to choose the hepatitis C virus protease inhibitor as a starting point? Are there any structural similarities at the active site or sequence similarities, etc.? Thank you.
Yeah, we had this idea about 2 years ago. These inhibitors, I looked at them, and they are peptidomimetic, so that's important. So they look like peptide chains with some unnatural substituents. The fact that they are covalent was an important consideration, because they can bind and covalently attach to the catalytic cysteine. I looked at the groups. Obviously, P1 group didn't seem to be right, but that's because in the hepatitis C protease, that site is quite hydrophobic, so you don't want anything there that makes hydrogen bonds. The P2 group, that fused cycle, seemed to be really perfect match for the SARS coronavirus 2 protease, because the S2 site is also hydrophobic. Then the P3 group is on the surface, so we really didn't care what that was. And P4 is hydrophobic, so we also knew that that site is hydrophobic. So we looked at these compounds in terms of first that they are peptidomimetic, so they mimic the substrate. They are covalent inhibitors, so they can bind covalently, and that improves the affinity, and that some of the groups looked like good match for the coronavirus protease subsites. So that's where we came from.
Did the meeting finish?
No.
I think Giovanna has to do this. I kept some. Okay, so we have a question from Juliette.
Hi, Andre. Actually, I had exactly the same or the similar question to Isai, because I was wondering why you started by looking at inhibitors of hepatitis C, and I was wondering if maybe the proteases of hepatitis C and the MPRO from COV2 had also a non-canonical triad like you presented today, but it seems that you mainly looked at it because of the structure of the inhibitor, right?
Yeah, structure of the inhibitor. The hepatitis C is more normal serine protease. It has a triad.
It has triad, okay.
Yes, it's not cysteine, it's serine, and structurally, it's actually a bit different. In terms of three-dimensional structure, it's actually completely different. There's nothing similar about the three-dimensional structure of the two proteins. The active site has some similarities in terms of binding subsites, but we mostly looked at that from the point of view of small molecules themselves. Yes.
Okay, because the KD of telaprevir for the SARS for the M protease you're studying was not very good, right, compared to hepatitis C protease?
Yes, of course.
When you started, yeah, okay.
one of the reasons, as I mentioned, is
Yeah.
because the P1 group is hydrophobic. If you don't have that hydrogen bond to histidine 163, that clearly kills affinity of the inhibitor to the main protease enzyme.
Yeah, thank you very much.
Jack, sorry, can I ask one more question in the same line?
Yes, of course.
Okay.
Thank you. Andre, do you think people are already working perhaps to combine the in silico screening? So do you think if we input all the viral protease inhibitors that are known into your computing algorithm, do you think this will be picked out? Like both the hepatitis C protease inhibitor will be picked out?
Yes,
as a
actually,
hit?
yes, that was done by some other group. I read a paper. They obviously did this, like you said, after the fact, after we and others published the ability of these inhibitors to bind to the main protease. They did their library screenings and the high throughput simulations, and they did pick up boceprevir, for example. It came out to be as one of the binders. So yes, it can be done. It was done.
Okay, that's a very powerful tool then. Yeah, thank you.
Mhm.
So we have a question from Raila.
Hello, Andrei, thank you very much. I have a question on the part of research where you did virtual screening of the so much amount of compounds, and you got only one active compound. And at the same time, you have mentioned that it's the proteases goes large conformational change while inhibitor binding. And my question is that, was it the limited step on your model search, and how your prediction and the X-ray data or neutron data is superimposed well?
Well, yeah, I can tell you that this simulation used multiple conformers, as I mentioned, of the protease, mainly from the PDB database with known structures. And that seemed to be a very important piece of this methodology. We know that others actually tried similar things at the beginning, and they only used the inhibitor-free structures. So they could never find really good compounds, because when these subsites are blocked, there's almost nothing that can bind. So this is so introducing a lot of conformations into the simulation was an important, and it is a limiting part of this methodology. It has to be included for success. Yes.
Okay, thank you.
Okay, thank you. I think we have no more questions, so we can thank again Andrei for these very interesting talks. And I thank you, all of you, for participating to the seminar, and have a good day.
Thank you, everybody.
Goodbye.